Oh, good afternoon. So today's video is a quick one on antibiotic treatments of microorganisms. So let's begin. Proteins account for more than 50% of the dry mass of most cells. They speed up chemical reactions. They are involved in defense, storage, transport, cellular communication, movement, and structural support. They are all constructed from the same set of 20 amino acids. Polypeptides are known as unbranched polymers built from amino acids, and a protein is classified as a biologically functional molecule that consists of one or more polypeptides. Amino acid monomers, these are amino acids which are organic molecules of amino and carboxyl groups, you can see on the right hand side, and these differ in their properties due to different side chains known as R groups. Amino acids are covalently linked by peptide bonds, so a polypeptide is a polymer of amino acids. Polypeptides range in length from a few to more than 1,000 monomers. Each polypeptide has a unique linear sequence of amino acids of carboxyl C N-terminus. So the specific activities of proteins results from the intricate three-dimensional structure. So a functional protein consists of one or more polypeptides precisely twisted, folded, and coiled into a unique shape. So a transparent protein shows the overall shape of the molecule and some internal details. A solid shape is used when structural details are not needed. There are four levels of protein structure: primary, secondary, tertiary, quaternary. Primary is a unique sequence of amino acids. Secondary consists of folds and coils in the polypeptide chain. Tertiary is determined by interactions among various side chains, R groups, and quaternary results when a protein consists of multiple polypeptide chains. The primary structure of a protein is its sequence of amino acids. Primary structure is like the orders of letter in a long word. It is determined by the inherited genetic information. With regards to secondary structure, the coils and folds of secondary structure result from hydrogen bonds between repeating constituents of a polypeptide backbone. These typically form an alpha helix and a folded beta structure of a beta pleated sheet. Tertiary structure is the overall shape of a polypeptide, which results from interactions between R groups rather than interactions between backbone constituents. These interactions result in hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals interactions. Strong covalent bonds called disulfide bridges may reinforce the protein structure. Quaternary structure results when two or more polypeptide chains form one macromolecule. Collagen is a fibrous protein consisting of 3 polypeptides coiled like a rope. Hemoglobin is a globular protein consisting of 4 polypeptides, 2 alpha and 2 beta subunits. The molecular components of translation involve a cell translates an mRNA message into a protein with the help of transfer RNA. Transfer RNAs transfer amino acids to the growing polypeptide in a ribosome. Translation is a complex process in terms of its biochemistry and mechanics. So the structure and function of transfer RNA. Each transfer RNA molecule enables translation of a given mRNA codon into a certain amino acid. Each carries a specific amino acid on one end, and each has an anticodon on the other. The anticodon base pairs with a complementary codon mRNA. A tRNA molecule consists of a single RNA strand that is about 18 nucleotides long. Because of hydrogen bonds, tRNA actually twists and folds into a three-dimensional molecule. Transfer RNA is roughly L-shaped with the 5 free ends that are both located near one end of the structure. The protruding free end acts as an attachment site for an amino acid. So here's a wee diagram showing you all that. With the codons and the anticodons, sets up a ribosome as a site of protein synthesis. The beginning with a 5N RNA for translation, free end at the end. So what is the genetic code? So there are 20 amino acids but only 4 nucleotide bases in DNA. What would happen if 1 or 2 nucleotides encoded for one amino acid? What would happen if 2 nucleotides encoded for another amino acid? What would happen if 3 nucleotides encoded for one amino acid? So basically 1 nucleotide is equal to 1 amino acids, meaning that in the first case there will only be 4, in the second case there will be 16, and in the third case there will be 64. So codons, triplets of nucleotides. The flow of information from gene to protein is based on a triplet code, a series of non-overlapping 3 nucleotide words. One of the two strands, two DNA strands, the template strand, provides a template for ordering a sequence of complementary nucleotides in mRNA transcript. The template strand is always the same strand for a given gene. The words of a gene are transcribed into complementary non-overlapping 3 nucleotide words of mRNA. During translation, the mRNA-based triplets called codons are read in a 5N to 3N direction. The non-template strand is called a coding strand because the nucleotides of this strand are identical to the codons. except that thymine is present in the DNA in place of uracil in the RNA. These words are then translated into a chain of amino acids, forming a polypeptide. Each codon specifies the amino acid, one of 20, to be placed at the corresponding position along a polypeptide. All 64 codons were deciphered in the early 1960s. Of the 64 triplets, 61 code for amino acids. 3 triplets are stop signals to end translation. Genetic code is redundant. More than one codon may specify a particular amino acid, but is not ambiguous. No codon specifies more than one amino acid. Codons must be read in the correct reading frame, correct groupings, in order for the specified polypeptide to be produced. Here you can see the triplets of nucleotides, the relative codons, etc. You can see the start codon AUG, methionine, and the stop codons UAA, UGA, UAG. So mRNA to polypeptides. So the first artificial mRNA polynucleotide synthesized with polyuracil. This is produced using the information consisting of a single amino acids of phenylalanine. And the conclusion is UUU is a codon for phenylalanine. tRNA carrying the phenylalanine would have AAA as its anticodon. So as mRNA molecules can potentially encode 3 different polypeptides, this depends on the frame used. However, you don't always know which DNA strand is being copied. So how many frames are possible? You have 6 possible reading frames. So this is an example. The fox and the dog ran off. The fox and the dog ran off. The hef oxa NDT. You know, you can see so only one makes sense. Same with the genes, only one reading frame makes a functional protein. You basically need punctuation to guide us, start and stop codons. So translation requires 2 steps. First, a correct match between a tRNA and an amino acid, which occurs via the enzyme amino-acyl tRNA synthesis, and the second, a correct match between a tRNA anticodon and mRNA anticodon. Flexible pairing at the third base of a codon is called a wobble and allows some tRNA to bind more than one codon. So ribosomes facilitate specific coupling of tRNA anticodons with mRNA codons synthesis. The 2 ribosomal subunits, large and small, are made of proteins and ribosomal RNA. Bacterial and eukaryotic ribosomes are somewhat similar but have different differences. Some antibiotic drugs specifically target bacterial ribosomes without harming eukaryotic ribosomes. So a ribosome has 3 binding sites for tRNA. T site holds the tRNA that carries the growing polypeptide chain. The A site holds the tRNA that carries the next amino acid to the added chain. E site is the exit site where discharged tRNAs leave the ribosome. Here's an example showing the E, P, and A sites, the large and small ribosomal subunit. Further example of how this all comes into the picture with the growing polypeptides and then the next amino acid to be added to the polypeptide chain within the P site, then the tRNA present as well. So building a polypeptide has 3 stages: initiation, elongation, termination. These all require protein factors in the translation process. Energy is also required for some steps. So the first step, initiation, the AUG codon, which codes for methionine, signals the start of translation. A small ribosomal subunit binds with the mRNA and a special initiator to transfer RNA. Then the small subunit moves along the mRNA until it reaches the start codon. Proteins called initiation factors bring in the large subunit that completes the translation initiation complex. Here's an example of that. You can see the small ribosomal subunit binding to the mRNA, and the large ribosomal subunit completing the initiation complex. During elongation, which is the second step, amino acids are added one by one to the 3 terminus of the growing chain. Each addition involves proteins called elongation factors. Elongation occurs in 3 steps: codon recognition, peptide bond formation, and translocation. Energy expenditure occurs in the first and third step. Translation proceeds along the mRNA in a 5' to 3' direction. The ribosome and mRNA move relative to each other, codon by codon. The elongation cycle takes less than a tenth of a second in bacteria. So you can see here the elongation stage and the codon recognition. And then the peptide formation here, and then the translocation. You can see the first step involves GTP, and then the first step also involves, the second step involves also GTP. With regards to termination, elongation continues until a stop codon in mRNA reaches the A site of the ribosome. So that's UAA, UAG, UGA. The A site accepts a protein called a release factor. The release factor causes the addition of a water molecule instead of an amino acid. This reaction releases the polypeptide and the translation assembly comes apart. So it's a bit more detail. Ribosome releases a stop code on mRNA, so it's UAG, UAA, or UGA. The release factor promotes hydrolysis. Ribosomal subunits and other components dissociate. So completing and targeting functional proteins. Often translation is not sufficient to make a functional protein. Polypeptide chains are modified after translation or targeted to specific sites in the cell. During its synthesis, a polypeptide chain begins to coil and fold spontaneously into a specific shape, a three-dimensional molecule, secondary and tertiary structure. A gene determines primary structure, and primary structure in turn determines shape. Post-translational modifications are also required before the protein can begin doing its particular job in the cell. So targeting polypeptides to specific locations. So two populations of ribosomes are evident in cells. Free ribosomes within the cytosol and bound ribosomes attached to the endoplasmic reticulum. Free ribosomes mostly synthesize proteins that function in the cytosol. Bound ribosomes make proteins of the endomembrane system and proteins that are secreted from the cell. Ribosomes are identical and can switch from free to bound. Polypeptide synthesis always begins in the cytosol. Synthesis finishes in the cytosol unless the polypeptide signals the ribosome to attach to the ER. Polypeptides destined for the ER or for secretion are marked by a peptide. A signal recognition particle binds the signal peptide. The signal recognition particle then escorts the ribosome to a receptor protein built into the ER membrane. A bit more of a teaching how this begins. Polypeptide synthesis begins. Signal recognition particle binds the signal peptide. Signal recognition particle binds the protein. Signal recognition particle detaches and polypeptide synthesis resumes. Signal cleaving enzyme cuts off the signal peptide, the signal peptide is removed, and a completed polypeptide falls into final conformation. There are differences between making multiple polypeptides in bacteria and eukaryotes. Multiple ribosomes can translate a single mRNA simultaneously, forming a polyribosome or polysome. Polyribosomes enable a cell to make copies of the polypeptide very quickly. So several ribosomes simultaneously translate one mRNA molecule, and in B you can see a large polyribosome in a bacterial cell, so it's much more simpler in that sense. So in eukaryotes, the nuclear envelope separates the processes of transcription and translation, and RNA undergoes processing before leaving the nucleus. So that's the end of this video today. Our next few videos will be on treatment of microorganisms and antibiotic resistance. And then after that, I don't know, I'll see which topics we have, but thanks very much for tuning in today.